So hello friends. Today we will discuss about the previous year questions. Okay, so it will be including all the NEET PG and FMG and NSET. Okay, so I will post the questions accordingly. So first we will discuss. So the malaria parasite was discovered by. So the options are A. Ronaldos and B is a Palmuller, C is a Laveron, and G is a Pampania. Okay, so the correct option here is C is a Laveron. So you want to remember these points. So first, the specific causative agent of malaria that is was discovered by Alphonse Laveron in the year 1880, and he found this parasite in the RBC, and he is a French army surgeon in Algeria. Okay, so just remember Alphonse Laveron discovered the parasite in the RBC. So here the additional point you want to remember here is Ronaldo's, and he discovered the sexual cycle of the parasite in the female Anomalous mosquito. Okay, fine. So Alphonse Laveron, he discovered the plasmodium in the RBC, whereas Ronaldo's he discovered the sexual cycle in the Anopheles mosquito. Second question. So the flagella not true is A. Locomotion, B. Attachment, C. Protein in nature, and D. It is an antigenic. So here the option is, the correct answer is, what is that? Attachment. Okay, so the flagella is not the organ for attachment. So for attachment it is in, what is that? Fimbriae. Okay, so here the flagella are the some of the important points, or the thread-like appendages which is protruding from cell wall. So just remember these are coming from the cell wall, and it has a protein called the flagellin, whereas fimbriae will be having a pilin. Okay, so it has three parts, that is a filament hook. Sorry, it's not hook, it is a hook, and it's a basal body. Okay, so and these are the parts of the flagella, and they are the organs for the locomotion. Okay, so which is usually used for the motility of the bacteria. So you want to remember the flagella, it is an organ for the locomotion, and it is an antigenic. Okay, and it is a protein in nature, except it is not for the attachment. For attachment it is in fimbriae. So third question, in nutrient agar the concentration of the agar is 1, 1.5, 3 and 4. The option here is 1.5. So here you won't remember many points in nutrient agar because it has been asked repeatedly in many exams. So first it is in, okay, so first it is in first it is a nutrient medium. Okay, so first it is a nutrient medium, and which is mainly for the non-fastidious bacteria. Okay, so nutrient agar, it is a nutrient medium for the non-fastidious bacteria, and it is a popular, that is, it is the most commonly used, because there can be a variety of bacteria can be grown in the nutrient agar as it contains many nutrients. Okay, you remember these points. Okay, so I have only highlighted the important points. Okay, so you must not leave any point over the explanation here. And what is made up of is, it is about 1.5% of agar and 0.5% peptone and 0.3% of an beef extract or the yeast extract and NaC. Okay, clear? Next. So next question is, so blood agar is an example of, yeah, it is an enriched medium. Okay, I think you'll be very thorough with this topic, right? So just remember, like, see what happens here is, generally the basal media, okay, we'll be adding a basal media to the nutrients, such as it can be a blood, or it can be an blood, or it can be a serum, or it can be an egg. So, and it usually supports the growth of the fastidious bacteria, whereas nutrient agar supports only for the non-fastidious bacteria, right? And here it supports the growth of the fastidious bacteria. And so what are the examples here is, first is a blood agar. So in blood agar you will be adding 5 to 10% of the sheep blood. Okay. So 5 to 10% of sheep blood is added to the nutrient agar. So the temperature here is 45. That you won't remember, because chocolate agar again it is for about 70. Okay. So it is used for the hemolytic property of the bacteria can be demonstrated. That is alpha hemolytic, beta hemolytic and gamma hemolytic. Okay. So just remember these blood agar will be, this is again a question. What is the percentage of the sheep blood? That is about 5 to 10%. Now chocolate agar, this is a heated blood agar which will be added to the molten nutrient agar at 70 degrees Celsius. Whereas blood agar is 45, here it is in 70. And here it supports for even H influenza. Okay, and here it is an important point. Chocolate agar is mainly for the H influenza. Okay, so next it is a lawless serum slope. It is mainly for the Corynebacterium diphtheriae. You won't remember this. And monophasic medium, it will be containing in one, that is in BHI, brain hot infusion broth, whereas bibasic medium it consists of two. That is, it consists of again a liquid phase, that is in broth, and then solid agar phase, that is in agar. So here the question is, example of enriched agar is blood agar. Another add upon points are the chocolate agar, low flow serum slope. So coming for next question, the enrichment medium for the cholera. A, it is in Venkatraman Ramakrishna medium. B, it is in thiosulfate citrate bile salt sucrose medium, and Carey Blair medium, and alkaline peptone water. So correct answer here is, yeah, it is an alkaline peptone water. So some words about that. Okay, so what is this enrichment broth is, it will be allowing the organism, that is a pathogenic organism, to grow, whereas it will inhibit the normal flora or the normal pathogens. So only the pathogenic organisms will be allowed to grow in this type of medium. And here the examples are, one is selenite F broth and tetrathionate broth. It is mainly used for salmonella and the shigella. And second point is alkaline peptone water. It is mainly for the Vibrio cholerae. Okay. So this have been asked again repeatedly in exam. Selenite F broth. They can give a history, like a baby or a child who is suffering from a typical stepladder fever and coated tongue, and he is having a bradycardia. So how will you proceed with the patient, and what culture you will be using for the organism? Then it will be in selenite F broth or the tetrathionate broth. Okay. Thank you, friends.